Thank you. Thank you all for joining me today. And thank you to Elizabeth for inviting me to speak. We were talking earlier about how my parents live just down the road in Doylestown. And so I've driven past what was then Delaware Valley College many a time on the way to the mall. So I'm excited to talk to you today about one of my favorite organisms, the fireflies. And I've been chasing the lights of fireflies for about 11 years now, since I started my PhD. I did my PhD at University of Georgia. And one thing that struck me as I was first starting to study them was that I thought I knew about fireflies, right? I went out as a kid, I caught fireflies, put them in jars or cut them in my hand. They're cute, easy to catch, they light up, people like them, right? But it turns out that behind all of this beautiful bioluminescence, there are darker themes. It's more complicated. There's things like deception, poison. So I just became so fascinated with these more hidden parts of firefly life. And so today I want to share a couple stories with you. So first I'm going to talk about firefly biology, so what firefly scientists have figured out so far. Then I'm going to share two stories with you about what I've learned in my quest for the reasons behind firefly light. And then finally, at the very end, I will give some suggestions for how you can actually help us study fireflies and help preserve them for future generations. I am going to be showing some videos, so if it's possible to be in a room where there, or to find a place where there isn't too much glare on your screen. If not, you can watch this back again later on the recording and watch it in a darkened room. All right, so I'm gonna play a video here. So most insects disgust people, right, in general, but fireflies are different, right? Fireflies have captivated human beings for a long time because of this beautiful bioluminescence that they do. So this video is taken in the complete darkness using a special camera, and it's taken in the forest down in the Smoky Mountains in June. And this species that you're seeing is Photinus carolinus, and it's a firefly that will synchronize. So you'll see them do a series of 6 or 7 flashes where they sort of start to flash together, and then there will be a period of darkness, like right now. And then they'll all start flashing together again. And so you can imagine being out in the woods just totally surrounded by all of these flashing lights. It's inspiring, and sites like this encourage us to sort of slow down, pause, and wonder at the natural world. So you may have, like me, been out encountering fireflies since you were a kid, right? Catching them in jars, or I've heard that some people smear them on their face to get the bioluminescent goo on their face. I prefer the jar method. But one thing that you might not know, even with all of this experience, is that despite their name, fireflies are not flies at all. And even though some people call them lightning bugs, they're not bugs either. They are beetles. So what does that mean? Well, a beetle is an insect. It has 3 pairs of legs.
I'm sorry, you can continue.
All right, 3 pairs of legs, right? So here we have our classic 3 pairs of legs insect here. Many insects have 2 pairs of wings, so things like your flies and your bees. In beetles, the first pair of wings has hardened, and it forms a pair of wing covers called elytra. So in this picture, the orange arrow is pointing towards the elytra, and then underneath that, you can see those flight wings. So this is characteristic of a beetle. Fireflies are beetles. But what makes a firefly different from other beetles? Well, in one case, all fireflies will bioluminesce as a larva. So here is an adult firefly where you have on on its abdomen a very well-developed light organ. Those two segments at the end are where the light is produced. Whoops, my picture is missing. Apparently I'm having some technical difficulties. I'll show this at the end. But a firefly larva will light up from two light organs on its abdomen. And again, I hope to show this again at the end. The other thing that is characteristic of fireflies, so they all light up when they're juveniles. They all have a head shield. So this is called a pronotum. Here's a picture of a resting firefly, and what you can see is that its eyes are entirely covered by this semicircular head shield. You have another beetle over here on the right that looks like a firefly, right? They look very, very similar. It's got the hardened wing covers of a beetle. It's got red and black on the pronotum. But if you notice, its head sticks out. It can't actually retract that head underneath the head shield. And so this is actually a soldier beetle. It is not a firefly. So the first fact that fireflies are beetles is usually something that's new to folks. But the second fact is that there's not just one species of firefly. There are actually over 2,000 species around the world, and recent estimates go up all the way to 10,000 different firefly species. So for a long time, our knowledge of where fireflies are was limited by personal observations, museum collections, and these days, right, we have our phones with us all the time, and so we've been able to gather more data about where fireflies occur using some popular apps like iNaturalist. So this is an app you can get on your phone and take a picture of the firefly that you see. That picture gets uploaded to a database that scientists actually review. And so we'll check to see which species it is, and then it gets logged in to this huge database of sightings around the world. So each of these dots represents a different sighting from a different person. And if you see a bigger dot, there are more sightings. And so what you should notice here is that fireflies are occurring all the way from Alaska down to Patagonia. So they're occurring throughout the world. They may look different based on where you are in the world. So here on this top picture, there's an ascosoma from South America. We have our characteristic Botinus pyralis from the US, Eastern US. And then on the bottom, we have a species from Romania. They all have the wing covers. They all have the head shield. And they're all fireflies from all the way around the world. So what this means is that your childhood experience, going out, catching fireflies, putting them in jars, is sort of a shared human experience. Fireflies have fascinated people around the world for centuries. So here's a small collection of examples of human fascination with fireflies. We have art from ancient China, literature from Japan. We've got songs from Europe. We've got books and tales from Africa. We've got things from modern America. We've got different cars and even Hollywood TV shows. So firefly, everybody loves fireflies, right? In North America, we have about 120 to 170 species, depending on who you ask. And there's a couple of main groups that I can talk about today. The first one to talk about are these Botinus fireflies. And one way to remember this, it's got a T in it, so they're sort of tiny, although some of them are bigger. And they are ones that generally you will be seeing in your backyards. A second group are Boturus fireflies, and this you can remember because there's an R in it, Boturus, and they're really big. So they have really long legs. They tend to have stripes on their elytra, on their wing covers, and they have a more pointy pronotum. There's also a group of fireflies called Pyractomina, and these tend to be a little bit broader if you catch them and look at them in your jar. And they tend to have stripes on the side of their pronotum. These other ones don't have that. There's also Pyrrhapiga, and this one is very, very small. It's totally black on the elytra. And then we also have Lucidota fireflies. So these are a bit bigger, and they have these antennae that are very long and sort of flattened and triangular. And I'll get to why they might have those in a bit. So if you're out catching fireflies, you get them in your jar, you look at them, you should be able to sort of tell which group they might be in. So aside from what they look like, fireflies have been studying, or scientists have been studying fireflies for a really long time because of these flashes that they make. And the flashes are actually mating signals. Each species has its own flash pattern. So it's another way that we identify them. And this pattern is sort of like, you can think of it as a Morse code that helps them to recognize, locate, and choose their mates. So I like this picture. This is my favorite field site in the North Georgia mountains. And I like this picture because it shows the flash patterns of at least 2 different species. So you have this yellow streaky one across the top that does a series of 6 or 7 flashes. That's that Photinus carolinus. And then you have this species in the background that's doing these sort of flashbulb flashes in the trees and on the ground. The flashes that you see in your backyard, and most of the flashes in that picture, are probably males that are patrolling for females. And so I'm showing you a schematic of what we think of as a flash pattern. The males, so in a single flashing species, the males will emit a pulse of light over, separated by an interval of darkness. Females, who are usually down in the grass or somewhere on the vegetation, will see the first flash, see the second flash, cue in on that interval, and then respond with a flash of their own after a species-specific delay. So the male will know when he last flashed. He'll see the female flash. He'll be able to tell the delay. And then if he likes what he sees, he'll go ahead and flash again at the female. And then she'll flash back, and they go back and forth and back and forth. We call this a flash dialogue. So here's a short video of a flash dialogue. This is a Mr. Mac Firefly, which you have around you. In the center on this stick is going to be a female, and then circled in pink. And then the male is going to sort of fly up in the back. And this is a double flashing species. So he's going to go flash, flash. All right, and so I'll play this video, and you should be able to see the male come. There's his first flash, there's his second flash, and she will rotate her abdomen and reply to him. He'll do flash, flash, and she'll rotate her abdomen and reply to him. And then he'll land, and we'll get a close-up. Flash, flash, she replies, and then they can find each other and mate. So she has to be able to tell his flash from all the other ones around. Females tend to like bigger and brighter flashes. This guy, this particular male, took about 20 minutes to find this female, so perhaps he was not quite the brightest. So in fireflies, it's really interesting. Females will choose the males, so they like bigger, brighter flashes, but males give gifts. So in some insects, the male will go out and say, catch some food, or bring some food, bring it back to the female that she can eat, right? And she can use that food to make more eggs. In other insects, males will actually catch the food, eat the food, and then throw it up so that the female can eat it, right? And that's the present that the male gives the female. In fireflies, the male transfers a gift called a nuptial gift during mating. So here's a picture of a female common eastern firefly on the left and a male common eastern firefly on the right. And he is transferring the nuptial gift that consists of both genetic material, but also nutrients. So things that she can use to make bigger, better eggs. You should be able to notice that there's a difference in the light organ between these two individuals, right? So the male has these 2 final segments. They're both yellow all the way around. Both of those segments will light up. And the female here, only one of the segments is really yellow. And just this little kind of rectangle in the middle, that's the only part that will light up. So that's sort of how you tell males and females in photinus. Remember, tiny photinus. So we know fireflies best for these flying flashes, right? But bioluminescence isn't just for adults. So bioluminescence actually occurs across the life cycle of the firefly. After mating, the female will go ahead and lay eggs on the order of 100 eggs, and they'll spend about 4 weeks, depending on temperature, as in the egg stage, and eggs will glow. After they hatch out of the egg, they are in larval form. So here's that larva picture that was supposed to pop up earlier. These larvae are active for up to 2 years, and they live sort of above and in the soil where they're hunting down earthworms and slugs and snails. They're eating some of your garden pests. Here in the lab, I'm showing you a picture of one eating an earthworm. So that's what we feed them in the lab. You can see these two little white parts at the bottom of the firefly larva. Those are the light organs of the firefly larva, so they will glow from that. And why does the firefly larva glow? Well, we think that this could be a warning. Fireflies are toxic. And so this glow is a signal, sort of like a neon sign to predators at night saying, hey, I am toxic, don't eat me. After their 1 to 2 years as a larva, they'll go ahead and pupate. And even as a pupa, they'll glow. So this is a pupa glowing in the lab. And depending on temperature, about 4 weeks as a pupa. And then they'll go ahead and eclose or hatch out into an adult. Not all adults light up. There are some species that light up as adults, and those are the ones that have that really well-developed light organ. But there are many other species that are actually unlighted. They lack light organs, and they come out during the daytime. So most species we think about, they are living only about 2 weeks as an adult. There are a few exceptions. During this time as an adult, they're not eating in general, they're just trying to find meat. So I told you before that females like bigger and brighter flashes. And so why don't fireflies just stay on all the time, right? Why aren't they just bigger and brighter? And in fact, why do some of them not use light at all? That should be an interesting question. Well, it turns out that those Photurus fireflies, those really big ones, they eat other fireflies. They practice something called aggressive mimicry. So a female Photurus, when she's first hatched out, she'll respond to males of her own species. So she looks for the specific flash pattern, and then she'll respond to that flash pattern. After she's mated, she turns into a femme fatale. What she will do is she will actually pretend to be a female of a photinus species generally, and she will look for photinus males signaling in the environment. And when she sees that, she'll reply with the female flash of the photinus. The photinus male will come in thinking that there's a photinus female there. You know, he's expecting her to be there, and instead he'll find this Photurus. And this Photurus female is not interested in mating with the photinus. She's interested in something very different, and she'll actually jump on him and eat him. So this can be sort of graphic. Here's a picture of some carnage after predation, and she's eaten up all his middle. So why the heck do they do this? Well, here we have our Photurus, are really big. All of these are tasty treats for a Photurus. Turns out, I told you that fireflies are toxic. All of these species that it eats have toxins, but Photurus seems to have lost the ability to make this toxin. And so we think that they are eating other fireflies to go ahead and gain that toxin, put it into their eggs, and protect their eggs from mammalian and amphibian predators. So in turn, if you're a prey species firefly, what are you supposed to do, right? There's a female mimicking your flash. Well, one hypothesis is that prey species might move their activity period so that they don't overlap with when those Photurus are out. And so these two species on the bottom, Pyrrhipiga and Lucidota, those ones have actually moved to the daytime. Those are unlighted fireflies. They don't use light anymore. Instead, we think they're using pheromones to communicate. This Photinus over here, we think has moved more towards sunset. So they come out right around dusk. Photurus tend to come out a little bit later. And so maybe this is an adaptation in response to that predation. So I've told you that fireflies differ in whether they use light or chemicals as adults. I've told you that they differ in the patterns of the flashes that they make, but they also differ in another important way that at least I can see on my screen, and that is color, right? So this train of 6 flashes is very yellow, whereas the ones in the background are very green. So fireflies actually vary in color from green all the way up to orange, just naturally. And some of you might be thinking, well, how do we measure color, right? And in fact, that's one of the projects that I did for my dissertation. So here is an undergraduate who worked with me, Jenna Palinch, and she is measuring the flash color of a firefly using specialized equipment called a portable spectrophotometer. So when the firefly flashes, the flash travels up this fiber optic cable into the machine, and it registers the spectrum or the color of the light. What does that look like? So here's a graph where I'm showing you the wavelength of light. This is the color of the light going from blue all the way up to red. And then on the vertical axis is intensity. So how bright is the flash at each of these colors? So here's one flash. What you can see is that the firefly flash isn't just, at 560 nanometers. It's not just green. And it actually has a range of colors. So it's a spread. But what we can do is we can fit a curve to this and extract the very tippy top of this to say this is the peak wavelength. And that's what we use as the color. So this firefly has a color or has a emission at 562 nanometers, which is yellow-green. That's how we classify that. And we can measure multiple flashes from a single individual. And what you should notice is that that peak emission wavelength stays the same for the same individual, right? So it's not varying a lot. So each individual has its own peak wavelength. And so we use this data to ask 2 questions. One is, why would signal color vary? And the other question was how? So what's the genetic basis of this? And so one hypothesis for why signal color might vary is that it might be an environmental pressure. So when you have a male firefly signaling to a female firefly, it's not in a vacuum, right? It's in the context of an environment. Say they're in a forest. If they're in a forest, the color of the flash really matters, right? Here I have a green flash and a yellow flash. One of these, the yellow one, is much easier to see against that green background ambient light. And if you think about it, it should really only matter when there is still light around, right? So firefly signaling in the pitch black, there's not going to be differences in the ambient light environment. But if it's, say, sunset, there's still a lot of sunlight sort of coming through the trees, that will create in a forest a very green ambient light environment. So it would be advantageous for a male to have a yellow flash to show up against that background, right? Females can see that yellow flash and respond. So if we look across species, some are active only in open field habitats. Some are active only in sort of closed forested habitats. Some are active in both or right at the edge between the two. And so you might expect there to be differences in the ambient light environment in which they're signaling. And fireflies, I also told you, differ in their activity time, right? Some come out during the day. We're not going to pay attention to those because those don't flash at all. But of the nighttime ones, some of them signal early in the evening around sunset, whereas other ones will only signal once it gets really, really dark, late at night. And so we can use this variation to ask, all right, let's go out, measure a bunch of different species and see, does signal color vary with the light environment in which they're signaling? So for my PhD, we chased fireflies across the eastern US, went to 52 different field sites over several years. And at each of these, we measured the flash color of at least 5 individuals per species per site. We also recorded the habitat, so open or closed, and the activity time. And we found that there is tremendous variation in color. So some of this was known before and other, it was new. So here I'm showing you how different firefly species are related. On the left, that's sort of, you can think of it as a tree of firefly species relationships. And on the right, I'm showing you the color, and each dot represents the color that we measured from a single individual. What you can notice is that there are some groups, like this Phausis or blue ghost firefly, they're all sort of green. And there are other groups where they're all sort of orange, like in Paracamina. And there are other groups like this Phatinus, where they're just all over the place, right? And even within one species, you can go all the way from green up to nearly orange. So the question is, does this correlate with habitat and signaling time? Well, if we look at late active species, and you look at the middle here, this is the mean of signal color, there's no significant differences between in flash color between fireflies active in open environments versus closed environments when they're active at the very, very pitch dark, right? But if you look at fireflies that are active earlier in the evening, ones that are active in closed environments, in forested environments with very green ambient light, have yellower flashes. So from this, we tend to think that signal color, this green to orange, is an adaptation to signaling environment. And it has to do with contrasting against the background. So my second question was, how does signal color vary? And this took advantage of some key biochemistry that we know about the firefly light reaction. So fireflies light up with the use of several different ingredients. So there's a luciferase protein with its substrate luciferins. These two act together with a little bit of energy, some magnesium and oxygen to produce light. So they put all these ingredients together, bam, light is produced. And we know this from key work done in the 50s by scientist Dr. McElroy at Johns Hopkins University. And here he is sitting with his pile of fireflies that he paid undergraduates to get. And he's used these fireflies to harvest their luciferase and study how the light reaction worked. We know a bunch about luciferase because since the 50s, we've used it in science to study things like energy use, but also to mark certain tissues. So you can put luciferase into certain tissue cells and look at how they spread, how they grow. And so we've used it to study cancer quite a bit. In nature, fireflies have one luciferase that they turn on in their light organ, and that's the one that they use in the light reaction. In the wild, if you take that luciferase out, you put it into a bacteria, you grow it up in a tube, and then you expose it to luciferin, it creates a green light. So this is in the wild. But what you can do is you can mutate different parts of it. So the protein is made up of different building blocks. And here's an example where they've changed building block 257 from one type to another. And you can see that you get stuff all the way into like the really red range just by changing that one building block. So the hypothesis, or the question here, is variation in luciferase sequence in those building blocks, is that responsible for the differences in signal color? So to look at this question, we focused in on one firefly species. This is the Big Dipper firefly, or the common eastern firefly. This is the blue states are where these fireflies have been sighted. And they do sort of a very characteristic J-shaped flash. And they're active right now. So we were out until 1 a.m. last night catching them. If you look across the populations of the species, so across all of these different states, and you look at the average signal color of each population, and these different populations are organized in terms of latitude. So Texas is at the very left. We've got New Jersey and Michigan on the very right. There's lots of variation in signal color among populations. And so if we go ahead and sequence the luciferase, get that sequence out of the firefly and look at it, what we would expect to see is a mutation somewhere that would match up with the color, right? This mutation is changing that building block, resulting in a color change. So that's exactly what I did. I did 16 individuals for each of 12 populations, sequenced all of their luciferases, and at the very end, I got out that all of the luciferases are identical. So this hypothesis did not hold, and it seems like luciferase sequence does not underlie color differences within a single species. And we still do not know how they regulate color. So this is an ongoing project. So I've told you about firefly biology. I've told you some stories about my own research as a graduate student. And I wanted to leave you with some information about Firefly Declines. When I am out giving talks, this is often a question that I get. And I often hear from people, you know, when I was younger, there were so many more fireflies. Are fireflies declining? And the answer to that is, for many species, we don't know. We are just starting to get to a point where there are studies starting to be published documenting declines in fireflies. So being able to monitor populations over years and get that data. We also are starting to get more information about, well, what are particular threats to fireflies? Is it habitat loss? Is it pesticides? What is contributing to their decline? And so we do have some really good evidence that preserving firefly habitat is important. So there are some species that are really widespread. Common eastern firefly being one of them. You'll see them in fields, you'll see them in forests, you'll see them in your backyard. But there are other firefly species that are limited to just one particular habitat. In fact, there's a species that is only found in salt marshes in Delaware. It's the only place it's ever been found. And that habitat has been threatened with development. And so once that habitat is developed, no more of that species. The other part of preserving firefly habitat is that light pollution is a huge deal for fireflies, as you might imagine, right? If there's lots of light around, their flashes can't be seen, right? And so turning off exterior lights during firefly season at night is a really excellent way to help with that. There's actually different degrees of light pollution that are bad to good. So, right, the worst might be a light that is just sort of broadcast and widely spread. But if you get the lights that direct the light down towards the ground, that's better because it's more localized. In fact, if you get sort of more warm colors, fireflies don't see red light very well. And so if you can get sort of more amber lights outside, that helps. In the lab, we use red lights to go ahead and look at fireflies when we're out studying them. And then another thing that you can do is help scientists to study fireflies. So we are at this age, right, where we can do citizen science. We can get your help you are out there, you're doing observations in your backyard or wherever you travel, we can use that data. And so there are two programs that I'd like to share. One, I'll put it up in the chat, the links. One is Firefly Watch, and this is run out of Massachusetts Audubon Society. And this is a program where you go out and you tell us when and where you've seen fireflies. And it's really helpful because it actually asks you to count how many fireflies that you have. So do you have like 1 firefly or 10 fireflies or over 100 fireflies? You don't have to be totally exact, but it gives us a little bit more information about abundance so we can start to document declines. And then the second one is this iNaturalist app, where you can log your sightings. You can take a picture of the firefly that you have seen and get that identified by a firefly biologist. So we're using this data to start studying where fireflies occur and how their abundance has changed over the years. And then I will put in a plug for World Firefly Day. So this started a few years ago. We have an international firefly organization called the Fireflies International Network, or FIN. And we gather every 4 years or so to have a big firefly conference. And this includes people who study fireflies in their backyard. It includes artists. It includes firefly biologists. And we all want to come together and think about firefly biology and conservation. And so World Firefly Day is coming up this year. It's on the 4th. And there will be videos posted to our YouTube channel. I'll put the link in the chat again. And these will include things like firefly poems. There's going to be a printmaking demo. There's going to be firefly videos from different people who study fireflies, take pictures of them, take videos of them. And so you're welcome to enjoy those things and celebrate fireflies for the amazing sense of wonder that they bring. So I will finish up by saying that science doesn't happen all by itself, and I have so many people that have helped me with both funding as well as with finding different places to collect fireflies. I also have my lab to thank. Here we are with all our red lights on, ready to lead some tours at the Pennsylvania Firefly Festival. So with that, I will leave you with a picture of my favorite firefly, the blue ghost firefly, and I will take any questions. Yeah, maybe like an inch long, maybe a little bit longer. They start off when they first hatch out, they're like 3 millimeters, super, super tiny. And we're not exactly sure what they eat then. Maybe they are eating other little worms in the soil. But once they get bigger, there are several that we think are specialists on earthworms. There are a couple aquatic species. So these are larvae that live in the water. Adults fly around in the air, but all of the larval time is spent in the water, and they are snail specialists. Bioluminescence has evolved 41 separate times, well, approximately 41, the number changes, but approximately 41 different times across the tree of life. So there's bioluminescent microorganisms. A lot of them live in the ocean. There's bioluminescent fish. There's bioluminescent mushrooms. There's bioluminescent, there was something about bioluminescence in sharks the other day. I guess that goes with the fish category. And so there's, all of these have a luciferase and a luciferin, but they're not the same genes. They're actually new genes that have evolved. The luciferase is the enzyme, the protein that is going to chop up the luciferin to create the light. We know that fireflies, in their genome, they have a gene that makes the luciferase. We don't know how they make luciferin. That's like the holy grail quest for photobiologists, is how do you make, or how does a firefly make that luciferin? So it really depends on what time in the season. So fireflies also, there's certain species that come out in the spring. There's ones that come out in early summer, ones in late summer. Right now, we're sort of transitioning from early summer to late summer species. And so you can kind of get both right now. So for example, last night we saw 1, 2, 3, 4 different species, starting from about 8:15 in the woods till when we left. But you might see more depending on where you live and the habitat you have. We were out in sort of a wooded with some mowed area, like a field. So one thing that would be really helpful is if people see these types of things, to go ahead and log them into Firefly Watch, log them into iNaturalist, or shoot me an email, because a lot of this is we can't be everywhere all at once, right? There's, you know, a handful of Firefly scientists in the US. We need your help.
Volunteers come out and use iNaturalist to help us collect all the biodiversity information that we can.
Right.
And we're actually doing a Firefly walk next Thursday. So we'll try to catch some of that for iNaturalist as well. You can go to our website at Wissahickon Trails and there's a registration there.
The best pictures for us to ID are when they're still. So like if you had them in like a little jar or on your hand, usually both one from the back, but also one from the front,
Right.
Would be helpful, because some of them, you know, you look at their sort of belly color to tell. Some of them we won't be able to tell to species just based on a picture, because the flash pattern is really important.
Right.
So if you are able to bring a stopwatch in the field with you, or just sort of estimate, you know, are they doing 2 flashes? Are they doing 3 separated by a longer interval? Are they doing 1 flash every 5 seconds? That is usually very helpful information. And I think some of that is asked in Firefly Watch.
Right.
Don't always match up, right?
Do they?
It's not like doubles always get a double back. We're not exactly sure. We think that the flash is under the control of the central nervous system, so they can change the type. So the type of flash they're making, for example, if you catch one, it's not going to do its patrol flash anymore, right? It's going to do more of an alarm flash or an angry flash.
Right.
But we're not exactly sure about the other genetics. Like, is there a gene for a single flash? Similar, and in places where they co-occur, they will hybridize. But for the most part, there's been something called reproductive character displacement, which basically means that fireflies that have similar signals, or fireflies have likely adapted to that to reduce conflict, and so they actually diversify their signals where they overlap. So you'll have species that have similar flash signals, but they won't be in the same area. They'll either be in a different habitat or they'll be in a different part of their range. And so they partition things out so nobody overlaps. You have to catch them. You can at a distance. It's not very good because as you go out, you're measuring more and more, right? If you just want to measure a very tiny light organ on a firefly, you have to get super close. And so we're actually pushing the fiber optic cable like right up to the light organ.
The other one.
Tourists tend to come out really late when they're doing their mating flash, but it's also when they're out earlier and they overlap with the prey species, you can't really see them very well because they're doing the reply of that prey species, right? And so what you'll generally see is that as soon as the Photuras start coming out, the prey species start going to bed for the night. Like they disappear. But where they overlap, then that's when you see a lot of the predation. Don't eat, we're thinking like 2 weeks as adults. For the Photuras, it might be 4 weeks if you feed them pretty well. There's one species of firefly, so the winter firefly, that might be 10 months as an adult. So these have the reverse schedule that you think of for a summer firefly. These ones actually emerge as adults in September around here, and then they will overwinter on the sides of trees, or over on the sides of trees, or just like in the first layer of soil. and they'll mate in the spring, lay eggs at the end of spring, and then they'll be gone in the summer. But their end of season is the summer, so they will die at the end of summer. Those eggs hatch out, they'll go through their 2-year larval cycle, and then they'll emerge in the fall of the year that they become adults.